Okay, so today I will give a brief introduction of the multimodal data integration in medicine. So today I will focus on two different parts. In the first part, I will give an introduction on multimodal data integration. For example, I will give some definitions about multimodal data. How can we integrate different modalities of the data? And then I will give an examples based on Alzheimer's disease detection. So let's move to the first part. You might want to know, what does multimodal data refers to? So the multimodal data refers to the data set that incorporate information from multiple sources and modalities, including medical imaging, genetic data, patient report outcomes, and clinical notes. So multimodal data integration is gaining popularity among industry and academia. So last month, Google DeepMind published a work related to multimodal data integration. So they introduced a multimodal data integration framework based on AI, which could consider 4 different types of the data. So we could see this is the data types they use. They use the report data, image data, video data, and signal data. So based on these 4 types of the data, then they make a decision, such as like if this patient have a specific disease, or could help the surgeon to assess the technical skills. Then I will give 2 examples to show how does the MedGimney work. So here on the left-hand side, you could see an X-ray image and the diagram from the user and the MedGimney. So the user wants to know, is there any anomaly in the X-ray image? Then we could see the MedGimney could give us some information and also the evidence why they make such a conclusion. And then on the right hand side, you could see some surgical imaging. So this surgical imaging is from the surgical video, and the user wants the MedGimney to help us to assess the technical skill of the surgeon. Then they give 3 different criteria and requirements to let the MedGimney to help us to assess if this video meets these requirements or the criteria or not. So the MedGimney could give us some response. So you could see here, they give some assessment based on some evidence. So this is there are 2 different examples to show how does MedGimney work. Then let's move to another part. So in medical world, we not only have signal imaging, we have a large amount of data we could use. So here I give some categories to show like which kind of data we could integrate in our framework to make prediction or to help us to assess the technical skill of the surgeon, or the or help us to do like personal medicine. Here are 4 different categories. So in the first category is an electro healthcare recording. So electro healthcare recording includes medical imaging, patient report outcome, and clinical notes. And also we could consider signal data from the wearable biosensor, so such as EEG, ECG, and EMG. Also, we could consider signal from ambient sensors, such as video or depth camera, the signal from microphones. And also we could consider the omic data. So we have different type of omic data, such as genetic data. So for genetic data, we have a whole genetic sequence and transcriptome, proteome, and metabolome, and antibody. So we have a large amount of data we could use in our framework to help us to do the precise medicine. So here are 4 different examples to show how can we use that in the real case scenario. So in graph A, here we have the image data and the genetic data. So here the input is a histology and the output is a genetic mutation. So here we extract some features from the histology and then we put them into, so for example, we could extract the features such as pathology of the tumor, and then we put into a framework and output the genetic mutations based on the histology extraction features. So this can help us to infer certain mutations from histology. And in graph B, so we could find the relationship between non-invasive and invasive modalities. Here is an example to show how can we find the relationship between the non-invasive and invasive modalities. On the left-hand side, you could see this is a CT. And on the right-hand side, this is a histology and the subtype of the histology. So we extract the features from the CT, such as the interest regions, and then we put into a framework and get the subtype of the histology. So this is the way how we could use a non-invasive image to generate the subtype of the invasive image. In graph C, we could uncover associations between clinical data and patient outcome. So what does it mean? So here you could see the histology on the upper side and the genetic segmentation on the lower side. So this is a histology. So we also could extract the features such as morphology of the tumor and inputs, and then we got this feature, and then we could get some features such as like the disease-associated SNPs from the genetic segmentation, and then we can fuse them and put into a framework, and then we can predict the risk of the specific regions in the histology. So it could help us to uncover the association between genetic data and histology. So here is another example how to integrate different data and implement that in the real case. So here we have the electric medical recording, such as the histology data of some patients. For example, the medicine histology, the medical history, and some laboratory test data. Then we have signal data from wearable devices, such as electrocardiogram. Then we fuse these features and do the risk prediction from the specific person and to give some results such as the treatment response and the resistance. So these are 4 different examples in the real case scenarios. After the general description of the multimodal data implementation and what does multimodal data means, let's move to some technical details about the multimodal data. So here I will show some multimodal data pre-processing techniques. So in multimodal data pre-processing, we could transform raw and unstructured data into a format optimized for model training. So what does it mean? It means that, for example, we have for example, X-ray image, MRI, text data, but if we put that into a machine, at that time, machine cannot understand what does it mean. So we need to translate the data into a form that makes sure the machine can understand it. So at that time, we could follow these 4 steps. So in the first step, we could do data cleaning. What does data cleaning help us to do? So in the first step, we could input missing values. And in the second step, we could remove duplicates from the raw data. So you might want to know why missing value occurs. Because, for example, in the laboratory test, the test was not ordered, or sometimes it's not sufficient for analysis. So we need to input the missing data. And sometimes for the vital signs, for example, the temperature, blood pressure, and heart rate. So in some time step, the value is missing. So at that time, maybe we need to input the missing value and make sure that all modalities of the data correspond with each other. And sometimes because of the system issues, we have duplicate records for the same patients. So at that time, we need to remove duplications from the raw data set. And then we should consider data reduction. So what can we do in data reduction and why we need to do that? Because, for example, we have a histology. So in the histology, the resolution is really high. If we put that image into a framework, then the computation complexity will really high. So at that time, we only pay attention to the region of interest in the medical imaging. So this is a way to do the data reduction based on medical imaging. Or sometimes we use genetic data. For genetic data, maybe we could consider like the whole genetic sequence, but it's a large amount of SNPs in the whole genetic sequence. So we could consider the disease-associated gene and then to extract the features from a specific segmentation, then the computation complexity will be lower. And for example, if we use the features from laboratory tests, there are a lot of features could be considered, but we cannot use all of them. So at that time, it's better for us to select the most significant features for us to input into a framework. So we call all of them feature selection. And sometimes we want to reduce the dimensionality of the data. For example, we have MRI. MRI is a 3D image, but sometimes we don't want to use 3D image. We only want to use 2D image. So at that time, maybe we could just use some of the slides in the 3D image and put them into the framework. So this is another way to reduce the data, so to do the data reduction. After that, we could do data integration. So data integration means that we create a unified data, merge the different modality of the data. For example, here we merge medical imaging, disease-associated genes, and laboratory tests. The reason why I do it, because we need to ensure the consistency among different modalities. After that, the final step is data transformation. So this step is quite important because we need to translate the data into the way that the machine can understand. So for the test data, we could use vector to represent the task. For example, we can use word to vector to represent a word, and then use a matrix to represent a sentence. And then we could use a vector to represent the genetic sequence, and we could do the rescale of the medical imaging, because sometimes the size of the image is different for different patients or in different hospitals. So at that time, we should rescale the medical imaging and make sure that the size of the image is the same, which could be input into a framework to learn. And also, we could consider data augmentation, because sometimes we don't have enough data to train the module. So we need to do the data augmentation to get much more data to help us to train the framework and get better results. Then we transfer it into a matrix and then put into a framework to learn the whole process. After data preprocessing, you might want to know, now we have the data, and how can we integrate that? So here I will show 3 different ways to integrate or fuse the data. So the first way is early fusion. So what does early fusion mean? It means that it refers to joint representation from raw data or features at the input level before feeding it to the module. So here I give you an example. This is a histology, this is MRI, and this is CT. So we extract the features from these three images and then fuse them. For example, we can use concatenation operation to fuse these three features together and then transfer into a metric and input into a framework or a deep learning framework, and finally get the prediction result. But for this, for early fusion, we need to know that we need to make sure all of the modalities should be similar, because here, even if the subtype of the image is different, because this is a histology, this is MRI, this is CT, but all of them belongs to medical imaging. So we should integrate the data from similar modalities if we want to use early fusion. And another choice is late fusion. So late fusion refers to decision level fusion, which trains a separate module for each modality and aggregates the prediction from individual modules for the final prediction. So what does it mean? Here I give another example. This is the genetic data. This is the medical imaging, and this is the tableau data. So we extract the features and give a prediction based on this module for genetic data. And we use another module to extract the features and give a prediction for the medical imaging data. And then we give the third module to extract the features and to give a prediction. Finally, we could fuse the prediction result based on, for example, we could use majority voting or averaging to get the final output. So this is another way to fuse a feature. For this kind of techniques, the late fusion, we could deal with heterogeneous data, because you can see here, this is the genetic data, this is medical imaging, this is tableau data. We don't need to make sure that all of the data belongs to the same modality. And we don't have the data synchronization limits. So this is an advantage of late fusion. And then here we could consider another ways to fuse the information, which called intermediate fusion. So what does intermediate fusion mean? Here we have the genetic data, we have medical imaging, we have tableau data. Then we could use different framework to extract different features and use another module to fuse the features. So the prediction can supervise the feature extraction module to improve feature representation under the multimodal context. So for example, here we could use, for example, fully connected layer to extract the features from genetic data. We could use, for example, the convolutional neural network to extract the features based on the medical imaging. And we could use fully connected layer to extract the features based on the tableau data. For example, some features from laboratory tests. And then we got embedded features from genetic, embedded features from medical imaging, and embedded features from tableau data. Then we could use another module to fuse them. For example, we could use attention mechanism to select the most important features and then fuse them, or we could use graph neural network to fuse all of the features, or we could use a contrastive learning to fuse the features. Finally, we could get a prediction result. So this is much more flexible, and it could help us to because the unified, so the unimodal embedding can be learned under the multimodal context. So because this is an end-to-end framework, and the feature extraction can be supervised by the prediction result. And also, it can tolerate the heterogeneous data and don't have data synchronization limits. So this is the advantage of the intermediate fusion. So after the introduction of the multimodal data integration techniques, we could move to an example. So this is an example based on Alzheimer's disease detection. So Alzheimer's disease is a progressive neurologic disorder that affects more than 850,000 people in the UK, costs the UK economy around 26 billion per year. And the damage initially appears to take place in the hippocampus, and then additionally parts of the brain can affect it and begin to shrink. And finally, the damage is widespread and the brain tissues have shrunk significantly. So here, I give an example. On the left-hand side, this is a healthy brain. And on the right-hand side, this is a severe Alzheimer's disease. For the clinician, the reliable diagnosis can be confirmed by complementary information among multimodalities, such as image, cerebrospinal fluid, and blood tests. But it is a challenge. It is difficult or challengeable for experts to recognize all shifts in all modalities for the Alzheimer's disease patients, leading to misdiagnosis. So we plan to create an automatic explainable Alzheimer's disease detection framework based on multimodal data. There are two different objectives of our project. The first one, we plan to precisely extract and combine features from each modality, including medical imaging, genetic data, and tableau data. Then we plan to figure out why the automatic framework makes such a decision, making the detection results trustworthy. So here, I will show some data types we use in the framework. So we use tableau data, imaging data, and genetic data. And I will give some details about what does tabular data mean, what does medical imaging mean, and genetic mean in this case. So here we could use structured clinical data such as, for example, the blood test, some indicators from the blood test, and for the balance test from the neurological exams, and the memory test for the cognitive assessment. So all of them could be embedded into a vector. Then we use medical imaging. So we use T1 3D MRI, but we do not input 3D image into the framework. We only use some of the slides. For example, we use the middle imaging from the 3D MRI, from the side wheel, from the top wheel, and from the front wheel. And then we consider the genetic data. So for the genetic data, we use SNPs, which is associated with the Alzheimer's disease. So for each SNP, we have non-allele, both alleles, and one allele. So at that time, we could represent the genetic data into a vector. After that, we could input them into a framework to learn 4 different types of the disease. So this is I think maybe we do not predict the type of disease, maybe it's a stage of the disease. So here we can see this is the genetic data, this is a medical imaging, and this is a tableau data. So we use 3 different frameworks to extract the features from different modalities. For genetic data, we use feature extractions. We use, for example, the Foley connector layer to extract the features, and then we put it here. And we extract the features based on a kind of convolution neural network, and then we put it there. And we use a Foley connector layer to extract the features and put it there. After that, we put all of the extracted features into the feature fusion network. Finally, we output the stage of the disease. So there are 4 different stages. The first one is normal control, so which means that patient is healthy. And then it's early mild cognitive impairment. So maybe you could consider it's stage 1. And then late cognitive impairment should be stage 2. And finally, is Alzheimer's disease. is stage 3. But we still have some challenges. So here I give 3 different challenges we have. The first one is that the feature association among different modalities should be explored because we use 3 modalities of the data. So we want to know the feature relationship between, for example, the imaging and genetic data, the imaging and tableau data. So the feature association among different modalities should be further explored. The second challenge is that the feature significance for each modality should be explored. For example, in medical imaging, so we want to know which region the machine pay more attention to when they make such a decision, or which SNP the machine pay attention to when they make such a decision. So we need to further explore about that. Then we also need to generate a human understandable explanation of the decision made by the machine. Because if we only give a heat map, then we cannot understand what it means. So sometimes we could output a human understandable language to let the patient understand why the machine makes such a decision. So this is everything I want to discuss today. Thanks for listening. If you have any questions, please do not hesitate to ask.
Thank you so much. That was such a great and clear introduction. Do we have any questions from the audience? Feel free to raise your hand or unmute yourself and go ahead and ask, or use the chat if you prefer, and I can read out your questions. In the meantime, ahead of our second talk, I will launch a poll that you can start filling out whenever, to give you a few more minutes to ask questions.
May I ask, please?
Yes, of course.
Thank you so much for the nice presentation. What I'm interested and also like to work usually is the explainable AI. So my question is, so when you expect that you will have an understandable or explainable AI, so are you going to use post-hoc explainable AI, or you are going to build the explainability within the model?
I think at the beginning we could use like framework explainable, such as we could combine the natural language with the explanation output. For example, if we have the heat map, then we could use some clinical report to translate the heat map information. So this kind of things could be considered as a framework explanation. And then we also could explain much more deeper, like we try to understand the parameters. So we train a lot of parameters in the middle, and then we could output some layers. For example, the heat map itself, we could do some techniques based on the heat map, or we could do some calculations based on the heat map, and then we could know, okay, this is a region and this is a relationship. For example, we delete one of the modality and output the heat map. So we could know, okay, the machine pay attention to this region. And if we add this modality, then we find out, okay, the machine pay attention to different region. So maybe this is another way to explain the mechanism of the machine. So I think this is, I think maybe there are different ways. So the first way could be considered as multitask learning. So the output is a language and the heat map. Another way is that we could try to delete some of the modalities and output the interested regions for the machine and try to learn the internal things in the deep learning neural network. So this is some ideas about that.
Yeah. Thank you. Good luck.
Hopefully it can help you to understand. Yeah.
Thank you.
All right, thank you for the question. We have another raised hand by Indra, so feel free to unmute yourself and go ahead and ask.
Hi, thank you for your presentation. I just have a quick question with respect to your fusion method. Now, I see that you do use, say, three modalities here, genetic, imaging, and tabular. So when you fuse the data, it's kind of like mandates that all the three modalities are available. So have you considered missing modalities? Say for a particular patient's data, you only have genetic and tabular. You don't have the imaging data. Or say you have only the imaging and the genetic data. So in those cases, how do you do the fusing? Because your model would have already been trained for, like, say, three modalities, but you're missing one of the modalities information.
Yeah, I think it's a good question. I think maybe we could consider graph neural network because graph neural network is a really flexible network. For example, if we miss some nodes, then it still can work, right? So we could consider graph neural network as a baseline and consider how can we embed this information into a graph, and then we could. So because graph is much more flexible, and if we miss some of the nodes, the framework still can work. So maybe we could try it.
That's interesting. Thank you.
Okay, there's another quick question in the chat that I want to bring up, which is asking basically about your rationale for using selected slides instead of 3D images, and maybe when that would be the case.
So which one? So this one?
I think it's referring to slide 12.
Okay, this
your
one.
Imaging data, I assume.
So here, this is the 3D image.
Yeah, so you mentioned that in some cases you would extract only selected slides from the slices from the imaging data.
Yeah. So we have a 3D image, but if we use 3D image as an input, so at that time the computation complexity will be really high. We don't want to train so many parameters. So at that time, we only use a middle image from the 3D MRI, from the front wheel, from the side wheel, and from the top wheel. So only 3 slices of the 2D image as the input. So is that clear?
Yes, I think that clarified it. There's another question in the chat that I will read out briefly while we're at it. So this one is asking if for your model, how much overlap is required in the sample data from different modalities to make good predictions, or if no overlap is required.
So what does overlap mean here?
Hi, I think it's my question. And by overlap, I mean one person provides, say, image data. Do you need the same person providing other data like text record or so basically, yeah.
If you want to use multimodal data integration, so if you want to use this techniques, maybe you should collect the tableau data, imaging data, and genetic data simultaneously. So which means that for one person, you have tableau data, imaging data, and genetic data. So
okay, so all data should be paired.
Yeah, all data should be paired. But sometimes some data is missing. Maybe you can impute the missing value based on some techniques.
Okay, thank you.
Yep. Any other questions?
Yes, there's two more raised hands in the, so I'll pass to Hui Xin Ng first because you've been raising your hand for a while.
No worries. Yeah, no, I just have a quick follow-up question because I related to the imaging data as well, because I know or remember from what you mentioned earlier is the histology data is usually super high resolution, so it could be very computationally intensive. And I'm wondering, and and also I think later you also mentioned that for certain kinds of data, you might want to do augmentation, and sometimes like clinical grade brain scans have kind of poor resolution. So I'm wondering, like, how do you balance the decision, like when to downsample or reduce the resolution or reduce the dimensionality, and when to augment? Maybe this is like a fairly broad question, but I'm curious, like, what your take is in this context.
Yeah, I think it's a good question because sometimes we need high resolution image, but we should consider the computation complexity. So at that time, maybe we could, for example, use a encoder to reduce the resolution and then consider it as a supervision for the high resolution image. For example, we use a lower resolution image as a supervision, and then we cut the high resolution image into different slices and then put into another framework. Then we could consider it as a sequence of the input. So at that time, we could deal with these issues, like can reduce these issues, I think.
Thank you.
No worries. Any other questions?
Okay, I think a final question from Halimat before we move on to the second part of the talk.
Okay.
Hi, thank you for your talk. I found it super interesting. So I have two questions. One was about, like, you managed to get imaging data, genetic, and tableau all in one, which is amazing because it can be so difficult to get different modalities of medical data. So it's like, what is the sample size and where are you getting those data sources? And the other question I had was, you're being strategic about selecting features that are associated with known Alzheimer's diseases, such as the SNPs and slices of the MRI scans. But are you not worried that this is biasing the potential of finding new, perhaps more useful biomarkers?
Yeah, I think these two questions are both interesting. For the second, I will answer the second question first. So for the second question, we still consider about that. So sometimes we select the features manually. So such as we select the SNPs based on our knowledge, and then select the image based on our knowledge. But sometimes bias occurs. So we try to use an automatic framework to extract the features and then to give some ideas like maybe this features is much more important, but we did not figure out it in the past time. So we could use a deep learning neural network or which kind of techniques to help us to generate the new biomarkers. So different from the manual selected features. And then for the first questions, I think there are several public data sets for Alzheimer's disease. There is a data set called ADNI, and you could find it. So in the data set, it includes medical imaging, genetic data, and tableau data. So you could also find other data sets related to Alzheimer's disease. But for other data sets, I think maybe it also has a similar data set as the Alzheimer's disease.
Okay, thanks so much. ADNI is a really great one, as you said. It is. So I was just wondering, do you have enough volume from that? Because I guess sometimes with the public data sets, the worry is that the samples are quite small.
Oh, yeah. Yeah, sometimes it's small, but I think the data sets that I mentioned before is large enough to deal with this problem.
Awesome, thank you.
No worries. I think another question, Cole?
Yeah, I think another question came up that we can take on if it's not too long. So go ahead, Samuel.
Hi, can you hear me okay?
Yeah,
Yes.
Great. That was there's really interesting slides, really interesting project. You mentioned the ADNI dataset, and I've been working with the ADNI dataset quite a lot. And one of the issues is that it's collected from lots of different sites across North America. And I was wondering if this is the data that you're using, whether you've considered any sort of harmonization process before you pass the data into the feature selection process.
So for these questions, I think for me, I just use a small number of the data. We do not use a large number of the data. So we just try the framework. So at that time, I haven't met that issue, but this is an issue we should consider in next stage, like because the data is selected in different, so even if it's in America, but it's from different regions or from different hospitals, so we should consider that challenges.
Yeah.
Thank you for your questions.
There's a very useful, there's sort of some quite useful tools around data harmonization that have been applied quite a lot in the ADNI dataset. So I'd be happy to share them if you're ever interested in that.
Yeah, it's quite good. Thank you.
Okay, great. Thank you for all the amazing questions. And yeah, happy for people to connect afterwards as well. So I will end the poll now and launch the results, share the results, while we move on to the second speaker. Can everyone see the results? Then I'll pass over to Hui Xin.
Okay. Hi, everyone. Just checking that everyone can see my slide, right? And not the presenter view. All right, I'm gonna get started then if folks can see. Okay, great. All right. So hi again, I'm Hui Xin. And today I'll be sharing a little bit about my research, and I'll also like do a code demo later, but they will all kind of like tie up together how it's related, like multimodal data, and we already touched a little bit upon explainability earlier from Yuhan's talk. So in any case, I am a PhD student at UC San Diego, and currently I'm a visiting researcher at UCL. And my research is focused on evaluating and comparing machine learning models that are used to predict a person's age. And I'm looking at in the context of bipolar disorder and seeing if there's advanced aging in this population compared to healthy individuals. So throughout my research, I've come to learn about interpretability and explainability methods, which I will refer to as XAI because it's quite a mouthful to say those two terms in full. And I'll also be talking about why they are especially important in the context of clinical applications of machine learning. And especially in my area when we are predicting the risks of cognitive dysfunction in neuropsychiatric disorders. All right, so here's an overview of my talk, and we're gonna zip through a range of topics that seem pretty disconnected from each other, but hopefully it will give you enough context to understand the motivations behind the eventual code demo that I'm going to do later with XAI methods. And I'll be using this library called Captain, and it's an open source extensible library for model interpretability built upon PyTorch. So with the increase in model complexity and also, of course, integration across data from different modalities, and the resulting lack of transparency of complex algorithms, XAI methods are increasingly important. And as a researcher, especially a researcher in clinical neuroscience, you want to find ways to understand what features are contributing to a model's predictions. And that's where XAI methods come in. So now to set the stage, we'll talk about age prediction models and its relevance to predicting illness progression in psychiatry. So I'm using my research area as an example here. We know from decades of research that aging and psychiatric disorders have a large impact on the brain, and that correlates with decreased mental and physical fitness. Neuroimaging methods have been promising and have been useful in terms of revealing phenotypes of structural and functional impact of pathology in the brain. And these figures that you're seeing now here are from a longitudinal analysis of neuroimaging data of bipolar disorder. It's from this consortium called the Enrichment Consortium, and it has data from over 20 plus sites across the world. And what they found here is there's a faster rate of cortical thinning in the areas highlighted in purple among people who have more mood episodes. So this reveals to us that there are shared features of aging and neuropsychiatric disorders, and this provides us the justification and the basis for developing machine learning models that predict age and to use that metric as a predictive marker of brain health across diverse populations. Now, the brain is complex, we know that, and we know that there are multivariate relationships between the brain features, and ultimately, some of these complex features and the features interactions are associated with dysfunction in neuropsychiatric disorders. And if we can capture those relationships, we can potentially predict the risk of eventual or further functional impairment. So in the context of bipolar, it manifests in symptoms like mania and depression, and it may put people at an advanced trajectory of aging as indicated by the red line here. Now, very quickly, let's get through how these age prediction, I guess like the predicted age is derived from the model. So there are 4 steps. First, we need to have a large training set of brain scans from healthy people. And then during the training process, the model learns how healthy brain scans look like across a large range of ages. And then we perform cross-validation, and eventually we apply to a target cohort of the population of interest. So what we end up with is this final metric, or the predicted metric, which is BrainPAT, and it stands for brain predicted age difference. And it's basically the predicted age minus the person's actual age. And a positive BrainPAT signifies that the person's brain is older looking compared to their chronological age. And a negative BrainPAT means we can regard that they have a younger looking brain relative to their chronological age. But having a marker with high potential for clinical utility is not enough. And we need to talk about how the model arrived at its decision. So this is where understanding model interpretability comes in. And why do we basically need to apply XAI methods to the model? And to further motivate the importance of XAI methods, I'll show you a simple example, which has nothing to do with brains, but it's kind of fun. So there's this rather famous example about how a neural network basically learned how to classify dogs versus wolves, but it actually didn't spot or learn the actual differences between the 2 animals. What it did instead was to recognize the backgrounds that these animals are in. And so basically, when you apply XAI methods to it, you actually see from the extracted image it's actually really relying on the snowy backgrounds to identify that animal. It's classifying that animal as a wolf as a result. Now, moving on, let's get into the definition of interpretability and explainability. So these 2 terms are often used interchangeably, but explainability is often regarded as a subset of interpretability. And interpretability is about generally and very broadly the ability to understand and make sense of the model's decision-making process. So in other words, we are asking the question of, can we understand how this model came to its decision? So as an example, we can say that a model is intrinsically interpretable if a person can just understand the internal workings of a model as a whole, like a decision tree, or at least parts of the model that are relevant to the prediction, like linear regression. And as for explainability, we ask the question of what is each node or layer semantically representing and how important it is to the model's eventual decision. And nested within explainability itself, there are two broad categories here. And the first is local methods that we can use to explain the model's decision for every single prediction. Basically, it's at the individual level. And global methods explain the model's overall behavior across the samples. So if there's a figure that I'd like you to walk away from today's presentation, it's really this schematic here that at least I personally found it was helpful when I was first learning about XAI, that this is probably the simplest way to think about the core concepts in XAI. But we'll also get into more details in a little bit. And basically, there are just further ways, a few more ways to categorize XAI methods. Now, with some foundational understanding of XAI methods, let's zoom out to the bigger picture of solving the age prediction task. So the decision that we make on what algorithm to use for training the training set involves like some trade-offs, right? And models in general can be divided into simple classical machine learning models and more complex algorithms like deep neural networks. And the first important criterion in model selection is, of course, accuracy. But in this case, and also honestly, in many cases, right, the simpler models have worse performance than the more complex ones. And on the other hand, we have this other criterion, which is interpretability. And what we have here is the simpler models are often more intrinsically interpretable, but the complex models are black boxes. So what we can do is apply these additional methods called post hoc explainability methods to extract explainability from these models. And I just want to take this opportunity here to reiterate that explainability is really a subset of interpretability in terms of the broader idea, because explanations are easy to derive from intrinsically interpretable models. And that'll just leave you with that message. But we'll move on and talk a little bit about what other factors we should consider when we're applying these XAI methods. And we have seen it from Yuhan's talk already that the data that we use for training can come in many forms. And the first question we should ask ourselves is, our data, is the data coming in the form of images or tabular data or time series data? And of course, can the method be applied to get the explain the model's overall behavior, or is it, can we also like get explanation for every single prediction? And here I want to use SHAP as a method, as an example here, because it's both applicable to global and local cases. And SHAP provides global explanations by ranking features based on their average impact on the model across all instances in the data set. So that's what we mean by getting the model's overall behavior. And for the case of for the local case, SHAP will systematically alter the values of each features while holding the other features constant, and that generates various combinations of feature values. So it then predicts the model output for each modified instance, and then it simulates the impact of changing individual feature values on the model's predictions. And the third point here is XAI methods can be model agnostic or model specific. So model agnostic simply means that it can be applied to any kind of machine learning model, and model specific means that it has certain constraints that it can only be applied to a certain class of models. And that is part of why you might have heard of SHAP or LIME, because they're popular because they are model agnostic. It doesn't mean that model-specific methods are inferior, they're just different. And there are some cases where you want to consider using model-specific XAI methods, because, so just to give an example, if you're working only with images and you're using a neural network for predictions, then CAM and GradCAM will be appropriate ways to obtain some form of explainability. Even though SHAP is popular, it might be too computational intensive when you have a highly complex neural network architecture with 1 million parameters. Now here's just a final slide to tie everything up, and we're going back to basically the first point I made, which is there's this trade-off about interpretability and performance. And when we get to models here, when you look at the x-axis, when you get to models on the left-hand side here with lower interpretability, we must be careful about how to interpret the results of post-hoc XAI methods. So for example, when we're applying SHAP, we should be aware that it captures both the individual contributions of the features and the interactions between other features as well. So when we are, I suppose when we are stating our results and interpreting them, we cannot just simply isolate the individual contributions because we run the risk of misinterpreting the importance of one single feature if we take it out of context. That's just to highlight SHAP is there. So we'll get into the code demo after I'm done with the slides, because I don't want to switch sharing different screens. But so very briefly, I'll just introduce the task here. We're going to use Captain Library to obtain the feature importance using different XAI methods. And the reason for picking Captain is it helps standardize interpretability methods across different projects, and also if multiple researchers are using it, then it also helps us standardize our results across the community. And it also, and then in this code demo, we'll also be using this library to apply to a simple 4-layer neural network. That's just to save computational cost and just make it simple for this tutorial purposes. And we'll train this neural network on a simulated data set with tabular neuroimaging features from individuals who are healthy and individuals with a bipolar diagnosis. And then we'll apply a few attribution methods or XAI methods to the model to extract feature importance. But I will not go into too much detail for each of these methods, since the documentation for them are quite comprehensive on the website itself. But I'd like to go through the results that you will see from running the code later. So you will be able to get this figure by running the code provided. But here's the main figure here. And on the x-axis, these are brain features. And on the y-axis, it's the feature importance from scale like negative to positive. And then what we see here in the blue bars are represented by, the blue bars are representing the gradient SHAP method. And gradient SHAP method uses gradients to estimate SHAP values over multiple per-trick samples, and it provides a more smooth and comprehensive understanding of the feature importance. And what we see here is it's pointing in a different direction compared to all these other methods. And I'm calling this out because in that line of code where we're running that function, it takes in the training data distribution as a baseline. So that means it takes into account how much each feature contributes to the deviation from the average prediction over the entire training data distribution. And in contrast, all the other methods that you see in red and the light blue, they are using zero baseline. So in that case, they are systematically setting each feature to zero so that when we're evaluating feature importance, we're evaluating it in a scenario where it's relatively a scenario where each single feature is actually absent from that data set. So that's why you see these like contrasting feature importance. And on the gray bars that you see here, these bars are representing the learned weights from the first layer of the 4-layer neural network. And it also kind of makes sense that they look very different from the other, the feature importance extracted from the other methods, because the weights from the first layer itself cannot fully capture the picture of overall feature importance, since the weights do get transformed across the 4 layers. Now let's zoom back out here and look at the big picture again and ask why this is all relevant to multimodal data integration as well. So multimodal data integration or fusion enables us to merge different data types, right? And increasingly, we have many libraries and tools for comparing different kinds of models. But ultimately, for practical usage, we need some form of explainability. By the way, I just want to give a shout out to Florence, who I believe is actually a Turing community scholar as well. So she's the author of this library, and I'm using her work as an example here. And in the context of neuroimaging, we have seen the advantage of leveraging the joint distribution of multiple data sets in enhancing our ability to differentiate between different kinds of psychiatric disorders. And of course, in age prediction, there are also increasingly more data generated across organ systems to create these aging clocks or age prediction models. And so this figure here is extracted from a paper that gives an overview of the different kinds of XI methods that are commonly applied to different data types across different organs. So if you're working on age prediction, I think this is a great paper to check out. So ultimately, I'd like to highlight that there is a lot of added value in applying XI methods to age prediction tasks, especially. But in this figure, I want to call out one I think especially important reason why we use XI methods, at least in the case for my specific research, where we're trying to predict cognitive impairment in bipolar, right? Is it does pave the path towards personalized interventions or personalized disease trajectory tracking, and personalized medicine is only possible if we can identify the early markers of age-related conditions and the targets for early treatment as well. So with that, I'm gonna end this presentation and get into the code demo. And I don't know, maybe we have a Q&A session in between, but thank you all for listening, and thank you, Sarah and Yuhan. Yeah, that's it.
Okay, let's see if there are any quick questions in the audience. Thank you so much. That was a very clear talk. And if there are no questions, I think you can go ahead with the demo, and then we can get questions again at the end.
Okay, that sounds great. So I believe, okay. So the code itself is all on the GitHub repository. And I would recommend that if you have a GitHub account, then you can fork this repository and then do git clone and then have it set up. But if you don't, there's an option to just go to analysis here and then download interpretability Jupyter notebooks and this module here and neural network brain age prediction, and also the synthetic data set, the simulated data set inside here, and then you can follow along. But otherwise, here I am just gonna briefly show, or I guess I briefly talk through what the code is. So here, what I have here, these are just the lines for installing the libraries that you would need to build the neural network. And the main library, the one library that you would use to get the XAI method is Captum, so it's included here. And it's fairly straightforward to use Captum. So once you have once you have installed the library and then imported it, and then you can choose what methods that you want to use based on your research question to apply to your model. So this is all just the imports. And then here we're reading the data frame or the data set itself. And then here, I basically implemented a very small or like simple case of doing grid search to optimize the hyperparameters. And eventually we train the model, and then we test the model on a data set of like healthy individuals. So I want to, instead of like you following me along like line by line to code it up, because this is actually really like a completed piece of code. I just wanted to talk through the logic of why I've made certain I guess decisions to write up the code in a certain way in the context of my research question. So again, here the goal here or the goal of my research here is to look at whether there's advanced aging in bipolar disorder, right? And ultimately, what we're predicting here is age. So one of the important things here is to make sure that the training and the test data's target variable, which is age, is actually has the same kind of like distribution. So the p-value of the t-test here of the group comparison should not be significantly different. So we are training, the model on a subset of healthy individuals and then testing it again on a subset of healthy individuals. And the idea here is that the MAE or MSE, whatever metric there, whatever loss function that you're defining, it should be as close as possible to 0 so that the model is accurate. But we're not testing the data, we're not testing the model on the individuals with bipolar in the sense that we're looking at evaluating the MAE, because what we're getting from there is the actual brain path that I mentioned earlier, which is there advanced is there like a greater difference between healthy individuals and people with bipolar? So the assumption is if the model learns what a healthy brain looks like, and there's a gap, we can take that gap as a definition or as a proxy for advanced brain aging. So this is all code for training and evaluating the model. Eventually, we get to the point where we are using the methods, the XAI methods from Captum. And again, it's like fairly straightforward. So the model is all defined before and above in the previous lines. And then I'm putting the model into the different kinds of XAI methods here as a argument. And in the talk earlier, I mentioned that one of the methods, gradient SHAP, uses the training set as baseline, and this is where you would define it. So if you decide to use completely 0 baseline, that's also possible, and you would just have to change the parameter here. And what you would get here is you generate the figure here. And as an exercise, actually, I thought that this is something we wouldn't be able to do right now. You could actually write your own code here by modifying these methods up here to get the feature importance for the bipolar group. Because that's also one of the research questions. I mean, that's a core research question we're interested in is which features is most contributive to this predictive, like this marker of brain health, which is the brain path. So I'm gonna pause here and see if there are questions.